DNA is a double helix composed of two antiparallel strands with a sugar phosphate backbone and nitrogenous bases, A, T, G, C, paired through hydrogen bonds. DNA replication was shown to be semi-conservative by the Michelson-Stahl experiment, which proved that each new DNA molecule contains one old strand and one newly synthesized strand. During DNA replication, several enzymes work together at the replication fork. Helicase unwinds the DNA, while single-stranded binding proteins keep the strands separated. Topoisomerase relieves the tension created during unwinding. Primase lays down short RNA primers because DNA polymerase cannot start replication on its own. DNA polymerase III then builds the new DNA strand in the 5-to-3 direction, forming a continuous leading strand and a discontinuous lagging strand made of Okazaki fragments. DNA polymerase I removes RNA primers and replaces them with DNA, and DNA ligase seals the fragments. However, the lagging strand's final primer at the very end cannot be replaced, because there is no available 3' end for polymerase to attach to. This causes chromosomes to shorten with each replication. 1 DNA polymerase I removes RNA primers used during DNA replication. Replaces them with DNA nucleotides. Also performs proofreading and fixes small errors. Most active on the lagging strand where many primers must be replaced. 2 DNA polymerase II. Mainly used for a final proofread. 3 DNA polymerase III. The primary enzyme for DNA replication in prokaryotes. Adds new nucleotides to the growing DNA strand in the 5 rightward arrow 3 direction. Builds the leading strand continuously. Builds the lagging strand in Okazaki fragments. Has proofreading ability to correct mistakes. Eukaryotic DNA is highly organized to fit into the nucleus. It wraps around proteins called histons to form nucleosomes, which coil into chromatin and eventually condense into chromosomes. Histons also play a role in gene expression. Loosely packed chromatin allows genes to be active, while tightly packed chromatin keeps them inactive. At the ends of chromosomes are telomeres, regions of repetitive DNA that protect genes from being lost during replication. Because the end primers cannot be replaced, telomeres shorten with age. The enzyme telomerase can rebuild telomeres, but it is active mostly in stem cells and cancer cells. The flow of genetic information follows the central dogma: DNA rightward arrow, RNA rightward arrow, protein. DNA and RNA differ in several ways. DNA contains deoxyribose sugar, is double-stranded, and uses thymine, while RNA contains ribose sugar, is single-stranded, and uses uracil instead of thymine. Transcription is the process by which a gene is copied into mRNA. RNA polymerase binds to the promoter, unwinds the DNA, and builds an mRNA strand using the template strand. After transcription, the mRNA undergoes modifications, a 5' cap is added, a poly-A tail is attached, and introns are removed by splicing. Students must understand how to switch between the template strand, used by RNA polymerase, the coding strand, matches mRNA except T rightward arrow U, and the final mRNA sequence. Translation is the process where ribosomes read the mRNA and build an amino acid chain. Initiation begins when the ribosome assembles around the start codon, OG. During elongation, tRNAs deliver amino acids that match each codon on the mRNA using anticodons. The amino acids link together to form a polypeptide. Translation ends at a stop codon, releasing the completed protein. Understanding codon charts is essential for converting mRNA to amino acids and switching between template strand, coding strand, mRNA, and protein sequence. Gene expression is controlled differently in prokaryotes and eukaryotes. Prokaryotes use operons. The lac operon is induced when lactose is present, turning the genes on. The TRP operon is repressed when tryptophan is abundant, turning the genes off. Eukaryotes use more complex regulation, including transcription factors, enhancers, silencers, and chromatin structure. CRISPR is a gene editing system originally from bacteria that allows scientists to cut and modify DNA at precise locations. Mutations occur when DNA is changed. Point mutations include substitution mutations like silent, amino acid unchanged, missense, different amino acid, and nonsense, early stop codon. Reading a DNA or mRNA sequence allows you to identify the type of mutation and predict its effect on the amino acid sequence. 1. Silent mutation. A base is changed, but the amino acid stays the same, because of redundant codons. No effect on the protein. 2. Missense mutation. A base change causes one amino acid to change. Can be harmless, harmful, or beneficial depending on the location. 3. Nonsense mutation. A base change creates a stop codon too early. Protein becomes too short, rightward arrow, usually non-functional. 4. Insertion mutation. One or more bases are added into the DNA sequence. Can cause a frame shift if not in multiples of 3. 5. Deletion mutation. One or more bases are removed from the DNA sequence. Also causes a frame shift if not in multiples of 3. 6. Frame shift mutation. Caused by insertions or deletions of bases not in groups of 3. Shifts the reading frame rightward arrow every codon after the mutation changes. The entry site for new tRNA molecules. A tRNA carrying an amino acid enters here and matches its anticodon with the mRNA codon. This is where each new amino acid is brought in during translation. 2. P site, peptidyl site. Holds the tRNA carrying the growing polypeptide chain. This is where peptide bonds form between amino acids. The chain grows longer each time a new amino acid from the A site is added. 3. E site, exit site. The exit site for empty tRNA molecules. After giving its amino acid to the chain, tRNA moves here before leaving the ribosome. Helps keep translation moving smoothly.